Hi, I'm David, and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. Figure 1.1 outlines ethanol fermentation and lactate fermentation. In ethanol fermentation, decarboxylation occurs when pyruvate is converted into ethanol. So, A is carbon dioxide and D is ethanol. Then, ethanol is reduced to ethanol. The hydrogen or electron comes from reduced NAD. So, B is reduced NAD and C is NAD. This also occurs in lactate fermentation when pyruvate is reduced to lactate. B wants you to explain how these processes enable cells to continue functioning in the absence of oxygen. Reduced NAD is oxidized in both processes to reduce ethanol and pyruvate. This recycles or produces NAD, so more are available. Glycolysis requires NAD. With the regeneration of NAD, it can continue. ATP can be produced via glycolysis despite the stopping of oxidative phosphorylation. ATP is produced by substrate-linked phosphorylation in glycolysis, where a phosphate group is directly transferred from a high-energy substrate molecule to ADP, resulting in the production of ATP. C. Describe and explain how rice plants are adapted to grow in folded fields. The command words describe and explain means that you only get a mark with one feature and its explanation. Therefore, you must provide three different features with the explanations in this case. First, there is parenchyma in the stems and leaves of rice plants. Large air spaces are found in the tissue. Parenchyma provides a low-resistance internal pathway, allowing oxygen to reach the root cells for aerobic respiration. Secondly, they have a high concentration of ethanol dehydrogenase. This enzyme allows the breakdown of ethanol. With a high concentration, they will exhibit high ethanol tolerance, so a high rate of ethanol fermentation will not be detrimental to the cells. They also grow very fast, so their leaves are out of water and exposed to more air. This allows them to have a more efficient gas exchange. There is a mark for AVP. For example, you can discuss the ridges on submerged leaves and shallow roots of the rice plants. Figure 2.1 shows the life cycle of a species of yeast. It can carry out both asexual and sexual reproduction. Mitosis occurs at both 1 and 3. In the budding process, identical daughter cells are formed. The chromosome number will not change. In 4, meiosis occurs. A diploid with two sets of chromosomes, A and alpha, divides into 4 daughter cells with only one set of chromosomes, either A or alpha. In meiosis, new genetic variation is produced. The random fusion of haploid cells at 2 also leads to genetic variation as it allows the combination of random genetic material. 1 and 5 both only show haploid cells. These are the cells produced in meiosis. They only have one set of chromosomes. 3 shows diploid cells as the haploid cells fused before this. B. Suggest and explain how the type of reproduction that makes spores during stage 4 is advantageous for S. cerevisiae in a changing environment. 4 is when meiosis occurs. This leads to genetic variation. 2 events in meiosis leads to variation, crossing over, and independent assortment. Variation refers to the differences between individuals of a species, such as in their physical characteristics, behavior, or genes. It causes some individuals to adapt better to a changing environment, allowing them to survive. Only those without beneficial features die. This prevents the whole population from being wiped out. Additionally, it allows stage 1 and 3 to occur. After 4, haploid cells are formed, and asexual reproduction in 1 can take place, following the fusion of the haploid cells. Mitosis can also occur in 3. Stage 4 causes the formation of genetically unidentical haploid cells. It allows for random fertilization or random fusion of gametes to occur. This leads to new combination of alleles in the diploid zygote. So, some individuals might have advantageous combinations of alleles. C wants you to suggest why a new harmful recessive mutation may not have a damaging effect on asexually reproducing populations of haploid and diploid populations of the species. haploid cells only have one set of chromosomes. This means they only have one copy of each gene. Each cell either has or does not have the harmful allele. Whichever cells with the harmful mutation will be affected or die. They help to remove the allele in the population. Therefore, the rest of the population will not have the mutation and will remain unaffected. In diploid cells, there are two copies of each gene. The recessive allele for the harmful mutation will not be expressed in heterozygotes. The dominant allele will mask it. So, individuals who have both alleles are unaffected by the mutation. There is a mark for AVP. You can state that the haploid cells help remove the harmful alleles from the population when the cells die. You can also talk about how the diploid cells that carry out asexual reproduction will not have any variation. So, there will not be a new combination of alleles in each generation. D. State two features of the kingdom fungi. It belongs to the domain Eukarya, so they are eukaryotic. You can also get this mark by describing an eukaryotic cell. They do not carry out photosynthesis. They are not autotrophic but heterotrophic. Many members are saprotrophic. They carry out extracellular digestion on decay, dead, or waste organic material. Some of them are parasitic. They can cause harm to their living host. The fungal cell wall is made out of chitin. It does not contain cellulose like plant cell walls. Most fungi have hyphae and mycelium. Hyphae are the basic cellular unit of filamentous fungal structures. A tangled mass of hyphae is called mycelium. Fungi contains multinucleated cells. This means each cell has more than one nucleus. Most fungi are multicellular. However, there are also unicellular fungi such as yeast. 3A wants you to outline the steps involved in the introduction of disease resistance to varieties of wheat. First, we have to select individuals with the desired characteristic to breed. In this case, we need to choose plants that are resistant to the disease. This can be done by exposing them to the disease. Their resistance and susceptibility will be shown. Then, breed the selected plants with resistance. Seeds will form following the pollination. We can now plant the seeds that have been produced. The offspring may show variation. Not all of them have resistance. We will expose them to the disease again and select resistant offspring. The selected individuals are bred together. This process is repeated for many generations. There is a mark for AVP. For example, you can name a wheat disease or talk about why sometimes outbreeding is required. B. Suggest two possible disadvantages of selective breeding. Selective breeding aims to produce a uniform population where all members are highly similar to one another. This causes a reduction in genetic variation. When we continually remove members with unwanted alleles, the gene pool becomes smaller. Eventually, there will be a decrease in heterozygosity and increase in homozygosity. In this process, we often repeatedly breed organisms that are similar to each other. This leads to less hybrid vigor. Hybrid vigor, also known as heterosis, is defined as the phenomenon where the hybrid exhibits phenotypic superiority over its parents. It leads to inbreeding depression as well. Inbreeding depression is a reduction in the fitness and survival ability of offspring that occurs when related individuals mate. When most members are homozygous for many genes, harmful recessive alleles have a higher chance of being expressed. The population is more susceptible to environmental change and new diseases, as all members have similar characteristics. If an environmental factors can kill one, it may also kill the others. There is a mark for AVP. You can provide an example of the consequence of inbreeding depression. A. Describe and explain how the use of a marker gene coding for a fluorescent product can show that the introduced gene of interest is being expressed in plants. A marker gene is a DNA sequence that helps identify successfully transformed host cells. It is used in the screening process to determine which cells have taken up the gene of interest. It is added together with the gene of interest. They are usually inserted near each other in the vector so that they can use the same promoter. Sharing the same promoter allows them to be transcribed and expressed together. When this happens, proteins coded by both genes are produced. The fluorescent protein enables the cells to glow under UV light. We will then place the cells under UV light to detect fluorescence. The presence of fluorescence indicates the transcription of both the marker genes and the gene of interest. We can now distinguish between the transformed cells and the ones that did not take up the gene of interest. You can talk about the benefits of using fluorescent genes as marker genes to get this AVP mark. B. Give 2 advantages of genetically engineered herbicide resistance in crop plants such as soybean. Herbicide-resistant plants are created so that they are not affected when herbicide is used to kill weeds on a farm. This can help to increase the productivity and quality of food. More food can be produced compared to the conventional farms. An increase in productivity will lead to a corresponding increase in income generated. Greater efficiency in food production can also reduce the cost associated with farm management. Herbicides can be used to kill weeds without harming the crops. When weeds are killed, there will be less competition between them and the crops for resources such as water and soil minerals. This also reduces and even eliminates the need to plow to kill the weeds. Less labor-intensive farming will make food production more cost-effective. C is about DNA microarray analysis. DNA hybridization occurs between the probe DNA and the added DNA due to the complementary base pairing. Hydrogen bonds will form between A and T and C and G. 2. Describe and explain how the level of expression of some genes can be identified using the DNA microarray analysis technique. To determine the expression of genes, we need to use the mRNA in the cells instead of the DNA. This is because when a gene is expressed, mRNA is produced through transcription. A higher level of expression means more mRNA is produced from this gene. After mRNA is obtained from the cell, we will make single-stranded cDNA using reverse transcriptase. A fluorescent tag or dye is attached to the cDNA. The DNA sample will then be loaded into the spots on the microarray plate. The hybridization of cDNA to probes takes place due to complementary base pairing. Then, the plate will be scanned using lasers or UV light scanners to check for fluorescence activity. The intensity of fluorescence indicates the level of gene expression. This is because greater expression causes more mRNA to be produced. Hence, more cDNA can be made from this mRNA. There will be more hybridization, leaving more fluorescent labels in the particular spots. Question 5 talks about the genes that control the body color and wing shape of the fruit fly. A wants you to calculate the expected number of each phenotype for a dihybrid cross, involving a heterozygous fly for both characteristics and a homozygous recessive fly for both. You can take a look at the genetic cross that I have done. The phenotypic ratio in this offspring should be 1:1:1:1. So, you can divide the total number of individuals in the observed values by 4 to get the expected values. In B, they use a chi-square test to compare the values. The calculated chi-square value is given. You are asked to assess the significance of the difference between the observed results and the expected numbers. The degree of freedom in a chi-square test is the number of categories minus 1. It is 3 because there are 4 phenotypes or categories. At p equals to 0.05 and the degree of freedom of 3, the critical chi-square value is 7.815. By comparing the calculated and critical values, we will know if we accept or reject the null hypothesis. Our calculated chi-square value is greater than the critical chi-square value at p equals to 0.05. This means the null hypothesis is rejected. A null hypothesis always says that there is no significant difference between the expected and the observed values. So, rejecting it means that there is a significant difference. A significant difference indicates the difference is non-negligible. It is not due to chance, as there is only less than 5% probability that the difference is due to chance. This means the difference is not random. There are important reasons behind. According to the table, the calculated value is greater than the critical values when p is equal to 0.01 and 0.001. You can also get a point by providing a more detailed conclusion using either of these probabilities. C. Suggest explanations for the observed results in Table 5.1. In the previous question, we found out that the observed result is significantly different from the expected value. We obtain the expected values by assuming the genes located on different autosomes. This means we are wrong about this assumption. The observed results shows that there are greater numbers of individuals with parental phenotypes and fewer numbers of recombinants. This shows that the two genes are linked. When two or more genes are found on the same autosomal chromosome, it is called autosomal linkage. The same allele combinations are inherited together from the parents to the offspring, resulting in the majority of the offspring having the same combinations of characteristics as the parent. In our case, dominant B is inherited with dominant D and recessive B with recessive D. There is no independent assortment since they are not on two different chromosomes. There are a small number of recombinants because of crossing over. Crossing over breaks the linkage, allowing some gametes to contain dominant B and recessive D, and recessive B with dominant D. It happens during prophase 1. There is a mark for AVP. For example, the genes are far enough apart on the same chromosome for recombinant to occur. Figure 6.1 shows the effect of glucagon on liver cells. The hormone binds to the receptor on the cell surface membrane. Their shapes are complementary to each other. The binding will activate a G protein. Then, the G protein will activate an enzyme called adenylate cyclase. It catalyzes the conversion of ATP into CAMP. CAMP is the second messenger. 2 wants you to state what happens to the rate of the processes when blood glucose concentration decreases. The glycogenolysis rate increases, allowing glucose molecules to be released into the blood. Glycogenesis decreases as glycogen formation from glucose further lowers the blood glucose level. Glycolysis decreases because cells will now use other respiratory substrates for respiration, as glucose concentration is low. Fatty acid synthesis decreases because lipids should be used to produce ATP at this moment. We do not want to use energy to produce more lipids when we do not have enough glucose. B. Explain the principles of operation of a test strip for glucose and suggest advantages for a person with type 2 diabetes of using biosensor instead of a test strip. A glucose test strip contains two immobilized enzymes, glucose oxidase and peroxidase. The strip is dipped into the urine sample. Glucose and oxygen in the urine react with glucose oxidase to produce hydrogen peroxide. The higher the glucose concentration, the more hydrogen peroxide is produced. Hydrogen peroxide will then react with peroxidase. Another substance called chromogen on the test strip will be oxidized during the reaction. This leads to a color change. A higher hydrogen peroxide concentration causes more chromogen to be oxidized, resulting in a darker color. The color is matched with a color chart to give an estimate of glucose concentration. A biosensor is quicker than a test strip. It gives an immediate reading. A biosensor is more sensitive. It can measure glucose in a sample even if the concentration is very low. The result is numerical or quantitative. Color matching is not required, so it is objective. Many biosensors can be connected to smartphones nowadays. The results can be stored electronically, allowing for further analysis. Lastly, a biosensor is reusable, while a test strip is single-use. A. Explain the need for many mitochondria in the presynaptic nodes and in the centromeres. You can get the first mark by stating the function of mitochondria. There are a few processes that require ATP in the presynaptic nodes and in the centromeres. First, the production of acetylcholine in the presynaptic nodes requires energy. Acetylcholine is the neurotransmitter used at the neuromuscular junction. Besides, energy is needed for the movement of vesicles toward the presynaptic membrane. The vesicles will then fuse with the cell surface membrane, leading to the exocytosis of acetylcholine into the synaptic cleft. In the centromere, ATP is needed for the active transport of calcium ion into the sarcoplasmic reticulum. This occurs after muscle contraction has taken place. Calcium ions are transported back to be stored in the sarcoplasmic reticulum. During muscle contraction, ATP binds to the myosin head, causing its detachment from the binding site on actin. This breaks the cross-bridge, allowing the myosin head to return to the original position. It is then ready for the formation of the new cross-bridge. There is a mark for AVP. Any correctly described event that needs ATP can be accepted. For example, sodium-potassium pumps on the presynaptic membrane restore the resting potential. After the exocytosis of acetylcholine, calcium ions are transported out from the presynaptic node. In B, we have a synapse between three neurons. Neuron X releases acetylcholine, causing an influx of sodium ions into the postsynaptic neuron. Neuron Y releases glutamate, leading to the influx of chloride ions. You are asked to explain if you agree with the statements made by a student. 1 is a correct statement. Sodium ions influx causes the inside of the membrane to become more positive. This leads to depolarization. Y is not true. Chloride ions are negatively charged. Their influx causes the inside of the membrane to become more negative. When it is more negative than the resting potential, hyperpolarization occurs. Since X is not stimulated, there is no sodium ion influx. The membrane is not depolarized. When both X and Y are stimulated, the influx of sodium and chloride ions will occur. The negative charge of chloride ions balances the positive charge of sodium ions, so the membrane will not be depolarized. C suggests the effect of multiple sclerosis on the transmission of action potentials in the affected neurons. The myelin sheath acts as an insulator, facilitating the rapid transmission of impulses. If they break down, the transmission speed will decrease. Fewer myelin sheath leads to a shorter local circuit. Local circuits involve the diffusion of ions within the axon, which help propagate nerve impulses by causing the next region to reach a threshold potential. Lastly, there will be less saltatory conduction when the myelin sheath breaks down. The propagation of impulses in an unmyelinated neuron is called continuous conduction. A. Outline the main features of non-cyclic photophosphorylation. Non-cyclic photophosphorylation involves both photosystem 1 and photosystem 2. Accessory pigments absorb light and pass the energy to the primary pigment. This causes the photoactivation of chlorophyll to occur in the reaction center of photosystem 2. Electrons are emitted from the reaction center in photosystem 2 and received by photosystem 1. Pigments in photosystem 1 absorb light energy, and electrons are emitted from the reaction center again. The electrons will then pass along the ETC. The electron carrier uses the energy lost by the electrons to set up a proton gradient. Chemiosmosis occurs and ATP is produced. The electrons emitted by photosystem 1 are used to reduce NADP. In other words, NADP is the final electron acceptor. The photolysis of water occurs. A molecule of water splits into oxygen, protons, and electrons. The electrons are used to replenish the one emitted from the reaction center. This process involves the oxygen evolving complex in photosystem 2, the OEC, also known as the water splitting complex, a water oxidizing enzyme. B. Name two intermediates in the Calvin cycle and state one example of the type of molecule that is produced. After carbon fixation, the 6-carbon intermediate splits into two molecules of GP. GP can be used to make amino acids, fatty acids, and lipids. GP may be reduced to TP by a reduced NADP. TP can be used to synthesize many different organic compounds, including carbohydrates, amino acids, and lipids. In C, we have a graph of the rate of carbon dioxide uptake against temperature. To calculate the mean increase in rate, we need to divide the difference between the 2 points by the temperature. The rates at 5 degrees Celsius and 20 degrees Celsius are 1.55 and 3.1. Dividing the difference between these values by the difference in temperature gives the mean increase in rate. 2. Suggest why the rate of carbon dioxide uptake levels off and then decreases after 20 degrees Celsius. Initially, the rate increases with rising temperature because it is a limiting factor. At different temperatures, the graph levels off because it is no longer a limiting factor. Other factors, such as carbon dioxide concentration, are now limiting the rate. High temperatures causes the plant to experience a lack of water, and this triggers the stomata to close. This mechanism prevents excessive water loss from plants through transpiration, as stomata are the main structure where water loss occurs. This will reduce the diffusion of carbon dioxide via stomata. If temperature continues to increase, it can cause the denaturation of enzymes. Enzymes such as Rubisco will denature, leading to a reduction in the need for carbon dioxide as the rate of photosynthesis decreases. There is a mark for AVP. For example, you can state the optimum temperature. A process called photophosphorylation may occur when stomata close during hot and dry conditions. It is a wasteful pathway that competes with the Kelvin cycle, as Rubisco acts on oxygen instead of carbon dioxide. On a cloudy day, the rate of photosynthesis will be lower, so the uptake of carbon dioxide will be reduced. 9a shows the changes in ADH concentration after a student drank 0.5 dm³ of water. You are asked to explain the results. Water is absorbed into our blood after it is consumed. This causes an increase in blood water potential. Osmoreceptors in the hypothalamus detect this change. The receptors expand, causing a decrease in the release of ADH into the blood. When there is a lower ADH concentration, fewer aquaporins are added to the cell surface membrane of collecting duct cells. Less water is reabsorbed from the collecting ducts, resulting in more water being excreted. So, there is a decrease in blood water potential. The excretion of water causing the blood water potential to return to the set point. This causes an increase in the release of ADH. More water is reabsorbed from the collecting ducts as there is less water in the blood now. This regulatory mechanism, which counteracts the stimulus to maintain a stable state, is called negative feedback. B. Outline the differences between the endocrine system and the nervous system. You must write for both sides to show the comparison. Do not only state the feature for one of them. Communication in the nervous system is carried out through an action potential. You can also refer to it as nerve impulses. A chemical signal called a hormone is used in the endocrine system. An action potential is a type of electrical signal. The nervous system also uses chemical signals known as neurotransmitters. The endocrine system only uses chemical signals. The mode of transmission for the nervous system is the neuron. Impulses are propagated in nerve cells. For the endocrine system, hormones are transported in the blood. The effectors are not the same. The response destination for the nervous system is the muscle, while the endocrine system targets specific organs, tissue, or cells. The transmission speed is faster for the nervous system and slower for the endocrine system. As a result, the nervous system responds more quickly. The location of the nervous system's effect is specific or localized. This is because the impulse is transmitted in a particular neuron to a specific effector. The endocrine system can have widespread effects. Hormones are released into the bloodstream, and the target cells with specific receptors may be located throughout the body. Lastly, the effects of nerve impulses are short-lived or temporary. For example, muscle contraction will only occur for a short time unless another action potential arrives. The impact of the endocrine system can be long-lasting or even permanent. For example, the effect of growth hormones is permanent and irreversible. In 10a, we have a passage about the mark release recapture method. After the animals are trapped, they can be marked by various methods such as waterproof dye, clipping fur, a collar, or electronic chips. The marking method depends on the structures and behavior of the animals. The period between the first and second captures must not be too long, as we do not want the population size to change during this time. Apart from emigration, immigration, and migration, we also do not want death or birth to occur, as this can affect the population size. The calculation we use to estimate the population size is called the Lincoln index. Like any other sampling method, replications are needed to increase reliability. More replications provide a larger sample size, allowing us to calculate a mean value which is more trustable. B talks about Himalayan balsam. You are asked to suggest why Himalayan balsam has been listed as an invasive alien species in the UK. An invasive alien species is a non-native organism from another region that threatens native wildlife and ecosystem. They have negative impacts on the environment or ecosystems. For example, they may cause ecological havoc in a habitat. They may also disrupt the food chain or food webs. This can lead to a decrease in biodiversity as some native species may be wiped out in the ecosystem. They can grow in large numbers and rapidly increase in population size. This is usually due to the absence of grazers, as they do not typically occupy this habitat. Invasive plant species can compete with native plant species for resources, such as minerals in the soil. Sometimes, they introduce new diseases to the ecosystem. This can lead to the death of many native organisms. They may also be toxic to animals. Some animals may consume them accidentally and die. Lastly, since they are flowering plants, they may attract pollinators away from the native species. A decline in pollination results in a decrease in the reproductive rate of native plant species.
That's all for today. If you think my videos are useful, don't forget to like, comment and subscribe. Thank you for watching and see you again soon.